Okay, next see that is about the restriction enzymes. So the restriction enzymes, that is the it is known as the nuclease enzyme. So this nuclease enzyme means it used to cut the DNA molecule. That is, the DNA molecule, it is cut through the phosphodiester bond. So here nuclease enzyme is mainly used to cut the DNA molecule. So the nuclease enzyme, these are mainly the two types. That is, one is the exonuclease and second is the endonuclease. Exonuclease and endonuclease. So see, that is here the exonuclease enzyme, it mainly cut the DNA molecule from the ends, that is from the 5' end or the 3' end. So cut DNA from ends, that is the from ends, that is the 5' or 3' end. Okay, while the endonuclease enzyme, this endonuclease enzyme, it mainly cut the DNA molecule at any point except the ends. म्हणजे ends सोडून, म्हणजे इथं हा DNA molecule आहे, ठीक आहे. It is the 5' end, it is the 3' end, it is the 5' end, it is the 3' end. So exonuclease enzyme, it cut the DNA molecule from this end or this end. Okay, while this endonuclease enzyme, it cut the DNA molecule from the point except ends, any point except ends. Okay, so it is about the nuclease enzyme, that is exonuclease and endonuclease. So in endonuclease enzyme, today we are going to see the most important enzyme, that is the restriction endonuclease enzyme, and this mainly used for the recombinant DNA technology. So next see, that is about the restriction enzyme, that is the restriction endonuclease enzyme. This restriction endonuclease enzyme, it mainly synthesizes in the bacterial cell. So see, that is here, suppose it is the bacterial cell, and see here it is the bacterial cell. In this bacterial cell, here is the circular DNA, that is its own DNA, and this DNA produce the restriction endonuclease enzyme. So when any virus that is infect on this bacterial cell, any bacteriophage for example, this bacteriophage infect on the bacterial cell, then this DNA enters into the bacterial cell. And then again this virus multiplies and large amount of virus are produced inside the bacterial cell, and this bacterial cell is killed down. But here the bacterial न्यूक्लिओसोम किंवा बॅक्टेरियल सेलमध्ये असणारं जे काही हे DNA जे आहे, दिस DNA प्रोड्यूस द endonuclease, that is the restriction endonuclease enzyme. And this restriction endonuclease enzyme, which is produced, it mainly kill or cut the DNA that is of the virus DNA. Okay, so it is the viral DNA. So this viral DNA, it is cut, that is by this restriction endonuclease enzyme, which is produced by this bacterial DNA. म्हणजे या बॅक्टेरियल DNA पासून जे रेस्ट्रिक्शन एंडोन्यूक्लियस एन्झाईम तयार होईल, दिस एन्झाईम इट कट द व्हायरल DNA आणि त्याचं कंप्लीट डिनॅच्युरेशन करून टाकतं. जर हा व्हायरल DNA या बॅक्टेरियल सेलमध्ये तसाच राहिला तर काय होतं, तर याचं मल्टिप्लिकेशन होतं आणि इतर लार्ज अमाऊंटमध्ये व्हायरस तयार होतात आणि ह्या बॅक्टेरियल सेल काय होतो, किल होतो. लक्षात घ्या. म्हणजे इथं रेस्ट्रिक्शन एंडोन्यूक्लियस एन्झाईम काय करतो, हा व्हायरल DNA जो आहे तो हा व्हायरल DNA कट करतो. ओके, डिस्ट्रॉय करून टाकतो. परंतु हा रेस्ट्रिक्शन एंडोन्यूक्लियस एन्झाईम इट डज नॉट किल द ऑर कट द दिस ओन DNA. Restriction endonuclease enzyme which are synthesized by the bacterial DNA. Okay, so this DNA is not cut by the restriction endonuclease enzyme. Why? Because there are the formation of the methylation on this bacterial group. That is, the bacterial DNA is produced, that is the methylation of the base, and these are mainly susceptible sites, that is methylation blocks the action of the enzyme. म्हणजे हा बॅक्टेरियल DNA जो आहे, सपोज इट इज द बॅक्टेरियल DNA. Okay, so here this DNA which produce the enzyme, that is this restriction enzyme. Okay, then here also the DNA molecule, it also produce the enzyme, that is the restriction enzyme. But here this restriction enzyme does not cut its own DNA. म्हणजे हा बॅक्टेरियल DNA जो आहे, हा बॅक्टेरियल DNA हा स्वतः इथं जे काही रेस्ट्रिक्शन एन्झाईम तयार होतं ते कट होत नाही. कारण का, दॅट इज देर आर द मिथिलेशन ग्रुप्स. Okay, so due to this methylation they are protected, that is this DNA is protected, and due to which here the restriction endonuclease enzyme does not cleave or cut the own DNA. Okay, so this enzyme only cut the viral DNA. Okay, so it is one most important thing. Then here the restriction endonuclease enzyme, this endonuclease enzyme, it mainly cut the DNA molecule in a particular sequence that is known as the recognition site. Particular site, that is, it is known as the recognition sites or the recognition sequence. Recognition site, or it is also known as the recognition sequence. So this particular sequence, it is also known as the palindrome sequence. It is also known as the palindrome sequence. So palindrome sequence or the recognition sequence means what? That is the sequence, if we read on the both strand, that is they are the same. म्हणजे दोन स्ट्रँड वरती, फॉर एक्झाम्पल सी हियर, दॅट इज इट इज द रेकग्निशन सिक्वेन्स. सो हियर इज द 5' एंड, हियर इज द 3' एंड. इफ वी रीड, that is AGCT, that is in this direction, that is through 5' to 3' end. Then in opposite direction also there are the similar sequence, that is AGCT. AGCT, here again AGCT. That is, if we read it, that is forward and backward, they are same. For example, that is, for example, see here one word, that is about the MADAM. If we read the spelling, that is MADAM forward, or it is the same if we read the backward, that is MADAM. It is nothing but the palindrome sequence. यालाच काय म्हटलं जातं, palindrome sequence. So here the restriction endonuclease enzyme, it cut the DNA molecule in a particular palindrome sequence, and when it cut the in particular palindrome sequence, then here the ends which are produced, ends these are produced, that is they may be the sticky ends or blunt ends. Blunt ends. Okay, so see, that is how they are sticky or the blunt end. So for example, see, that is here the one restriction enzyme, that is here the ALU1. It is the enzyme, that is the restriction endonuclease enzyme, that is the ALU1, and it cut the DNA molecule that is here in this sequence, that is it is the palindrome sequence like this. So here it cut this like this. So here the two ends are produced, that is the one is the A G like this, and another is the T C like this. So here another, that is the C T, and here the A G. So these two ends are the blunt ends. These are the blunt ends. Then next, that is here the another, that is the BAM3. It is the restriction endonuclease enzyme, and this restriction endonuclease enzyme, it cut the DNA molecule, that is like this here from this end and here from this. So here the ends which are produced, that is one end is here the only the G, that is the guanine, and remaining part like this, that is the G A T C C. Okay, then here the another part here remain, that is the G guanine like this, and here the cut, that is here is the G A, then T, then C C. Okay, so here cut, that is G A T C C. Okay, G A T C C like this. So this here, these are the sticky ends. These are the sticky ends. Okay, so like this here, the cut of the DNA molecule. So here, so here foreign DNA, that is it mainly joined to the sticky end. These are the sticky ends. या पद्धतीनं हे स्टिकी एंड्स इथं हे तयार होतात. ओके, इट इज द ब्लंट एंड. इथं मात्र फॉरेन DNA जॉईन होऊ शकत नाही, पण हे अनपेअर्ड नायट्रोजन बेसिस, हे जे अनपेअर्ड नायट्रोजन बेसिस आहेत की याच्यावरती काय होतं तर फॉरेन DNA हा काय होतो, जॉईन होतो. सो हियर देर इज द फॉर्मेशन ऑफ द स्टिकी एंड्स. लाईक हियर ऑल्सो द इको R1, हियर इज द अनदर restriction endonuclease enzyme. This restriction endonuclease enzyme, it mainly cut the DNA molecule, that is here forming the end, that is the G guanine, and here also. So there is the formation of the sticky ends, that is again here the guanine remains as it is. So here the cytosine pairing like this. So here the sticky end, that is the produce, that is the AATTC. So here AATTC. Okay, and here is the guanine. Here also there are the AATTC like this. Okay, so these are the two sticky ends. These are the sticky ends. So see, that is here the restriction endonuclease enzyme, it cut the DNA molecule in a palindrome sequence. म्हणजे तो DNA molecule काय करतो तर हा रेस्ट्रिक्शन एंडोन्यूक्लियस एन्झाईम रिकग्नाइज करतो. म्हणजे जेव्हा हा DNA मॉलिक्यूलवरती रन होईल, म्हणजे समजा आता हा DNA मॉलिक्यूल असा आहे, ठीक आहे, आणि इथं नायट्रोजन बेसिस असे भरपूर आहेत, परंतु जिथं रेस्ट्रिक्शन पॅलिंड्रोम सिक्वेन्स असेल किंवा त्याला आपण काय म्हटलेलं आहे रिकग्निशन साईट म्हणू. सो सी दॅट इज हिअर, सपोज हिअर इज द रिकग्निशन साईट, देन हिअर वेन दिस run, it cut the DNA molecule only in the recognition site. So for example here G, A, T, T, C. So here is the recognition site. Then here G, A, T, T, C. It is the palindrome sequence. Okay. So here when the enzyme run along this DNA molecule, then it recognizes the palindrome sequence and then cut like this. It cut the DNA molecule like this and produce the sticky ends. Okay. So here in some cases, that is the enzyme, some enzyme, that is for example ALU1, this enzyme when cut the DNA molecule, it produce the blunt end. So while another enzyme, that is BAM3 or EcoR1, when cut the DNA molecule, they are produce the sticky ends. आणि जेव्हा sticky ends तयार होतात, these are mainly used to manipulate or incorporate, that is for the foreign DNA. Okay, so it is about the restriction endonuclease enzyme.
Restriction enzymes. In this module you will learn about restriction enzymes. Restriction enzymes are enzymes that recognize specific sequences within the DNA and cut the DNA at the restriction site. Thus, they are also called molecular scissors. Werner Arber, Hamilton Smith, and Daniel Nathans were awarded the Nobel Prize for Medicine in 1978 for discovering the restriction enzymes. Recombinant DNA technology owes its development to the discovery of restriction enzymes. Restriction enzymes were first discovered when it was found that some bacteria have a defense mechanism and are able to prevent the multiplication of invading viruses or bacteriophages. These bacteria selectively cut or restrict the foreign DNA but not the self-DNA. The enzymes which cleave the foreign DNA were named restriction enzymes. Self-DNA is modified, methylated by the host's modification enzyme, whereas the foreign DNA is unmethylated and so is cleaved by the restriction enzyme. Hundreds of restriction enzymes have been isolated and purified from different species of bacteria. Restriction enzymes belong to the group of enzymes called nucleases. Nucleases are enzymes that catalyze hydrolysis of the DNA. These may either be endonucleases or exonucleases. Endonucleases cut nucleotides within the DNA, whereas exonucleases removes nucleotides from the ends of DNA. Restriction enzymes are endonucleases. After studying how type 1 and 3 recognizes the site, let us check the different step that type 2 uses. The type 2 restriction enzymes recognize and bind to a specific DNA sequence which may be 4 to 8 base pairs long and cut the sugar phosphate backbone of both the strands that make up the DNA double helix, giving rise to fragments of specific sizes. Since type 2 restriction enzymes cut the DNA in a precise and reproducible manner, they are used for gene cloning. Type 2 restriction enzymes are used for gene cloning. That's interesting. Don't you think it should be covered in detail? Restriction site for the type 2 restriction enzyme have a twofold rotational symmetry, that is, it is palindromic. It reads the same in both strands of DNA, read in 5' to 3' direction. Each restriction enzyme recognizes a particular restriction site, but different restriction enzymes recognize and cut the DNA at different restriction sites. However, some type 2 restriction enzymes recognize the same restriction site. Such enzymes are called isoschizomers. For instance, the restriction enzymes SST1 and SAC1 recognize the site GAG-CTC. Moving on the next series of restriction enzymes. These enzymes give rise to fragments with staggered or cohesive ends. Such fragments have single-stranded overhangs at both ends of each fragment. ECOR1 recognizes the site GAA-TTC and cuts the DNA within the site between G and A on both the strands to give rise to two fragments with staggered ends. The single strand overhangs can base pair with each other or with other DNA fragments having complementary ends. The restriction enzymes that generate fragments with cohesive ends are useful in gene cloning. Thus, it can be said that restriction enzymes are used for isolation and cloning of specific DNA fragments and for the construction of restriction maps. A restriction map is a diagrammatic representation of a DNA molecule showing the restriction sites of the different restriction enzymes. In this module, you have learned restriction enzymes are the molecular scissors that are a part of the restriction system in bacteria. Restriction enzymes are endonucleases. Restriction enzymes are named with specific reference to the bacteria from which they are isolated. The first letter comes from the genus name and the next two letters from the species. The next alphabet indicates the strain used, and lastly, Roman numeral signifies the order of discovery of the enzyme. Restriction endonucleases are of three types: type 1, type 2 and type 3. Types 1 and 3 cut the DNA outside the restriction site. Type 2 recognize a specific sequence which may be 4 to 8 base pairs long and cut the DNA within this site, giving rise to fragments of specific sizes. It is used for gene cloning. Restriction site for the type 2 restriction enzyme has a twofold rotational symmetry, that is, it is palindromic. Some restriction enzymes cut both the strands of DNA at the opposing phosphodiester bonds to give rise to blunt end fragments. Some restriction enzymes give rise to fragments with staggered or cohesive ends.